Good morning. Thanks for coming. This is a special day for us, and we're happy to share it for you. I'm Phil Needleman. I'm the retired interim president of the Danforth Center. I'll just briefly tell you, because the main event is for you to see the new president. How do you recruit a president? We had a search committee process which was pretty unique. As with others, at least 7 search firms were interviewed. The search, of course, was led by Roy Vagelos, the former CEO of Merck and the former head of biochemistry. I can tell you there's no search like working with Roy. The search committee then interviewed the candidates, the 4 candidate search companies, and we picked one. We had him here on the same day that at lunch we asked Roger Beachy, who is now going off to Washington, recruited to be the president of the National Institute of Food and Agriculture, to lay out his vision. The unusual hooker is identifying at least 12 of the leading plant scientists in the world to be our advisors. And about 8 of them came to St. Louis, some were on the phone, and they were each asked to spell out their vision of what plant sciences could be. And they were also coached, make up your mind in advance and have your list of 3 leading candidates. And that generated, and then we tapped into who are the leaders in plant sciences and academia. Before that, the board and the search committee really committed to the notion that we want the strongest possible scientist who is also a splendid administrator and people person. So we had rigorous criteria, almost unforgivable, including being a member of the National Academy of Science. But that doesn't imply that you could be a great administrator. Well, the search goes on, candidates are identified, the list is winning down, and then Roy Vagelos spends an hour or an hour and a half with all of those 18 consultants getting our list down to the finalists. In addition, the faculty here, highly energized to have an input, then identified independently, and their advice had to be unanimous, and they identified the candidate. And it's nice when the search and the insight people agree. Well, the candidate who you know is Jim Carrington. Really represents something very important, because we believe plant sciences at the Institute in St. Louis in terms of academia and industry is really at a great transition point. It's not just the solid science underway, it's the explosion of genomic data, which is now low cost, which is generating billions and terabytes of data, which brings out the marriage of computational science and plant science, and the application of these tools so you can change whole systems of plants for the benefit of the world and fulfilling the mission of the Plant Sciences Center. The one other thing I'll say is I'm an old road runner, so I was chairman in a medical school, I ran big pharma, I've searched for all kinds of scientists, heads of departments, deans, and heads of companies. I've never seen a single candidate who so fit the moment and the ambitions and the place of the science. So this search was a pleasure, and you'll see why we think Jim Carrington is so unique. Let me introduce Bill Danforth, who is truly a patron saint of St. Louis and plant sciences. Understand it was Bill. I learned when I was meeting Bob Thatch at Washington U. It was Bill, when young Thatch was becoming the head of biology, who said in response to a question, what should we build? It was Bill who said, you know, you ought to consider plant sciences. And Thatch recruited Roger Beachy to Washington University. It's Bill who conceived this institute. It's Bill who runs the Plant and Life Sciences Coalition and brought together the garden, many universities, Monsanto to this. So please let's welcome Bill Danforth.
Thanks, Bill, very much, and thanks for taking on the job of interim president. And Bill has been interim president for about a year, but his time hasn't stopped since Jim Carrington will not be here full time until the 1st of May. But we all owe a great debt of thanks to Phil Needleman, and I'd just like to ask the Phil Needleman advisors, admirers, the Phil, advisors too, but admirers of Phil Needleman, to clap your hands. Phil has described the search process, and we came out with Jim Carrington. Jim Carrington is a very noted scientist, a member of the National Academy of Sciences, who has made a special impact on small RNA or silencing RNA, which is a very important new field of development. And he brings great strengths to us that fit in well with everything else that's going on in St. Louis, with what's going on at Washington University and Missouri Botanical Garden, University of Missouri St. Louis, and other institutions in our community. We think it's a perfect fit, and one of our goals has always been to build St. Louis as the world capital for plant science. Our other missions, in addition to that, are to help improve human nutrition, help end starvation, and to help preserve and enhance the environment. So those are our goals, and the way we can contribute to those goals is through absolutely first-rate science. Jim Carrington was educated at the University of California, his undergraduate degree at Irvine and his graduate degree at Berkeley. He has since then worked at the Texas A&M. He's worked at Washington State University, and currently he's at Oregon State University in Corvallis, where he holds a named professorship and is also in charge of a large research project, a large research group consisting of about 90 people that is involved in understanding genetic information and applying mathematical techniques better to understand genetic information and handle the vast array of data that comes from it. We are also impressed with his vision and his ability to work well with people and identify great scientists to work with him. So with that, I introduce you to our new president, who will assume his full-time duties on May 1st, Jim Carrington.
Thank you very much, Phil. That's a wonderful introduction. And thank you, Phil, not only for the comments, but also for your service. Hopefully your continued service until spring. I would, Dr. Danforth, when you were talking about Phil, I thought you were going to say, would all the admirers of Phil please stand up? I'm tremendously honored to be here. This is a very humbling experience. The fact that you all showed up, not only the press, but the center staff, the students, the postdoctorals, the scientists, and the administrative staff. This is very humbling to be standing in front of you. The legacy of this center has already been laid down, has already been established, and My goal is to preserve what's already been done, preserve that legacy that was established by Dr. Danforth, that was executed by the founding president, Roger Beachy, and that was carried on and in fact extended in this interim period by Dr. Needleman. And again, I really want to express appreciation for the vision and the leadership that's preceded me, particularly Dr. Danforth and the Board of Trustees, which is the most supportive group of people for an organization that I've ever worked with. It's really an honor to be working with all of you here. The reason that I became interested in this position when asked to consider it and ultimately agreed to do it was because this is an entirely unique center. It's an entirely unique organization in the world. We have the opportunity to combine great fundamental science as it relates to plants, to figure out how plants work at a detailed level, a meaningful level, and in scientific circles, a very high impact, at a very high impact level. But that's not what makes the Danforth Center unique. What makes it unique is the commitment and the mission to take that fundamental information and apply it to where the needs are in the world. And there are many needs, as you well know. Malnutrition is a serious problem around the world. Food that is available may not be nutritious enough. We have serious environmental problems. We have a dependence on energy that's not sustainable, and it needs to be converted to a renewable energy system. And it has to be done not at some vague point in the future, but it has to be done quickly. The center is uniquely positioned to directly address those problems. The scientific staff that is here, from the students, technicians, postdoctoral, up to the group leaders, the members, they're committed to achieve this. And I'm honored to join it. I'm honored to share with you, for those who have questions, some ideas and some the way we perhaps see the future moving. And again, thank you for the opportunity. I'd be happy to answer any questions anyone might have.
Dr. Carrington, with your background in gene silencing and computation, what do you hope to do to take the Danforth Center forward? What will your general area of emphasis be?
The general area of emphasis may not change noticeably in the near future. Where we do see some potential for early impact is in filling some of the slots that we can accommodate with new scientists. That is going to enable us to make some hires at the principal investigator member level that will allow us to address some needs in technology. As Dr. Danforth and Dr. Needleman mentioned, we're in the genomic era, which means we have the potential to not only acquire vast amounts of genomic data, but also information about all cellular components. We do parts of that very well here already. There's other parts of that that we can do, that we can expand. We can apply new computing technology, new computing approaches, the integration of excellent biology, computation, quantitative science, even physics and chemistry. Merging those fields together in a systems-oriented approach is really where I think a big impact can be made early. So my research program will move here, so I'll have a lab, I'll be one of the members, and we'll continue that part of it. But I really look forward to integrating what we're doing in my laboratory with other things, other programs going on at the center.
And just follow up, with your ability to interpret all the data that's being dumped into this field, what sort of breakthroughs could you achieve that would the layman could understand?
Mm-hmm. Yeah, that's a very good question. So let's rephrase this a bit and ask, what are the big questions in plant science that need to be addressed, and what are the big problems that need to be solved? I like to think big and then ask what we can do to achieve those big goals. The big questions in plant science include things like, why do plants grow where they do? Why do some plants grow better with a little bit of water and others need vast amounts of water? What is the nature of the chemicals that plants make? There's a wonderful program here led by Tony Kuchan looking at medicinal chemistry that is entirely plant-based. We know just a fraction of the chemistry that goes on in plants. Furthermore, we have the technology now to extend work that before could only be done using very well-characterized so-called model plants. We can extend the technology to lots of plants, including food crops, energy crops, medicinal crops, and others. So these are some of the areas on the basic plant science I think we can make a big impact. Now, applying that knowledge to where the needs are, I think most of us can understand quite well. Improving the nutritional quality of food. There's a tremendous program here focused on cassava. Cassava is a staple crop for a large portion, particularly the underdeveloped world. It's a poorly, the nutritional quality of cassava is very poor. The spoilage rate of cassava is much too high. The protein content of cassava is much too low to provide a sustainable diet. Now, these are some of the real important outlets for the basic science that's done here, to translate that science to where it's needed. And that's the unique aspect of the center, is we're positioned, we've got not only the facilities and the infrastructure, the scientific staff, the administrative staff, we can do this. All right, well, I'm going to turn the microphone back over to Phil, and thank you very much for coming. I appreciate the opportunity.
Jim, Bill, and I will be available if you have more questions you want to ask individually. I think we look forward with great excitement. I would come back to say the third part of our vision was making an impact on St. Louis and the world. The level of commitment to computational biology and genomics really greatly enhances what we could do here, what will be done at Washington University in plant sciences, what will be done in Monsanto and other universities. So we see this as kind of part of the dream that St. Louis will be the focus for plant sciences in this country, complemented with the ability to move discoveries into applications and even to start new company ventures. So thank you all for coming, and we'll be available for additional questions as you'd like. Thanks.